So good morning to all. So we welcome you on the lecture number 20 of our Gyan course. So today lecture will be delivered by Professor Kavita ma'am, and the topic is about precision therapy exploiting synthetic lethality and case study. So thank you.
So I told you about synthetic lethality before. So today I'm going to give you 4 cases of synthetic lethality. Okay, and you guys know that very well. So for example, if there are 2 pathways for the same outcome, you delete one, the cells will use the other pathway and they will remain viable. Either A or B, you can delete either of them and the cells remain viable, but if you delete both of them, the cells they die. So what happens in cancer cells, often one pathway is already defective, so they entirely rely on the other pathway, but normal cells have both A and B pathway. So if you inhibit the other pathway, normal cells will survive, cancer cells will die. Okay, so that is that. This is the same thing shown here. So these are called cancer vulnerability. We are targeting cancer's vulnerability because it is addicted to one pathway, while normal cells have two ways to achieve the same outcome. Okay, so this is exactly here shown here. Non-malignant cells, they have 2 ways to attain cellular viability. So either you delete this or gene A or gene B, it remains viable. But in tumor cells, one is already defective. So if you inhibit the other one, they die. Okay, and normal cells live. So you guys know about BRCA1, BRCA2 and PARP, but I'll briefly tell you once again and then we'll discuss 4 other cases. So what are the functions of BRCA1 and BRCA2? They are tumor suppressor proteins and they are involved in DNA repair pathway. Okay, so DNAs are broken in 2 ways, single-stranded break and double-stranded break. Double-stranded breaks are obviously much more complex and much more dangerous. So these guys are involved in repairing the double-stranded breaks, okay, by a process called homologous recombination or HR. That is the function of BRCA1 and BRCA2, and they ensure DNA replication happens with minimal errors. DNA is our hereditary material, so cells make all efforts to make sure there are no mutations introduced. Okay, so now PARP1 is also a DNA repair enzyme that identifies and repairs DNA damage, primarily single-stranded. Okay, so if you inhibit PARP, then BRCA1 and BRCA2 takes over and they will repair it. Okay, so PARP under normal circumstances will repair single-stranded DNA. BRCA1, BRCA2 will repair double-stranded DNA. But if you inhibit PARP, BRCA1, BRCA2 will take care of both single-stranded and double-stranded. If you inhibit BRCA1, BRCA2, PARP will take care of both repairs. So they compensate for each other. But in many cancers, BRCA1, BRCA2 are deleted because they are tumor suppressors. So cancer cells have a habit of getting rid of tumor suppressors. Okay, so they get rid of BRCA1, BRCA2, then the cells are entirely dependent on PARP to repair DNA damage. Okay, and in that case, if you inhibit PARP, normal cells don't care because they have BRCA1, BRCA2, but tumor cells will die. That is synthetic lethality. Okay, so this is shown here. Single-stranded breaks are fixed by PARP, and if you add PARP inhibitors, then this single-stranded thing will not be fixed. Eventually, this will lead to double-stranded break. So normal cells will fix it because they have BRCA1, BRCA2, but cancer cells will die. Okay, so just to give you an idea, in breast cancer, 50 to 60% of breast tumors have BRCA1 deficiency. 40 to 55% have BRCA2 deficiency. Males have very little deficiency of BRCA1 in the breast cancer. Okay, pancreatic cancer 1 to 3%, both male and female. Ovarian cancer, again huge percentage, and prostate cancer also some percentage. Okay, so at BRCA1 and BRCA2, they are on different chromosomes here. Okay, so BRCA1, BRCA genes and So BRCA1, BRCA2 deletion is pretty common in cancer. Okay, and when people have BRCA, yeah, go ahead.
So, this PARP enzyme, so when a single strand breaks, is it the diester bond that breaks, or which one breaks, and then how it is ligated? Which one is broken?
So there are many ways for DNA damage to occur. So sometimes what happens that this is the whole strand of DNA, right? So phosphodiester bond remains intact. The purine ring will be hydrolyzed. So it's like a missing tooth. The gum is there, the tooth is missing, right? So purines are removed very easily, 5,000 times per day per cell in our body. Pyrimidines are 500 times per day per cell. So just removing the base, the glycosidic bond is hydrolyzed. That's very easy. But the phosphodiester bond is intact. Okay. So in that case, what they do base excision repair. So they cut basically the gum from both sides just exactly. So this is missing just exactly next to it, fix it with a new base using the second one. In case of PARP, it is the phosphodiester linkage which is broken. So it could be one or two pieces, it could be a bigger piece. So in that case, they do nucleotide excision repair. So they cut out about 12 to 13 base completely along with the gum, right? Completely, then the other strand is there. So that acts as a template, and DNA polymerase II comes in and fills it, and then it is ligated. So that's how it works. Yeah.
Previous prostate cancer, can it affect women?
Prostate is unique to men, ovarian is unique to women, and in here men also get breast cancer. So only 1 to 2% have deletion of BRCA1, up to 9% of BRCA2. But prostate is obviously only for men, ovarian. Let's see the higher percentage. So women who have BRCA1, BRCA2 deficiency, they are prone to both breast cancer and ovarian cancer. That's how that actress Angelina Jolie, she had this ovarian fallopian tubes all removed. Okay, so this is a very easy test to get it done and be preemptive about it. Okay, so this is really there are environmental risk also with impaired BRCA1, BRCA2. Because why environmental risk? Because environmental can break your DNA, right? If you're exposed to wrong things, chemicals or extremely bright sunlight for very long, these all things lead to DNA damage. BRCA1, BRCA2 are absent. That is a very serious thing. Okay. And even long bouts of being sedentary correlates with lower BRCA1 gene expression. Okay, so when BRCA1, BRCA2 is lost, then homologous recombination repair fails. They cannot fix double-stranded breaks. Cancer cells become dependent on backup repair pathway, and that is where PARP-mediated things come. So that is why PARP, hey, no cell phone. Okay, no cell phone in my class. Okay, so PARP inhibitors, so if you use PARP inhibitors like olaparib, then DNA damage accumulates and the cells will die. And normal cells will live. Why? Because they have the backup pathway. Okay, so that is the whole idea of that. So these are FDA approved PARP inhibitors. Okay, and they are extremely useful. Okay. Now let's look at second case for synthetic lethality, KRAS and TBK1. So yesterday we spent a lot of time on KRAS. KRAS is one of the most mutated gene in humans, and it is responsible for vast majority of cancer, especially highly aggressive cancer. Now here is the thing, when these oncogenes promote cancer, they mess up the physiological signaling pathways to an extent that they become highly dependent on alternate pathways. Like somebody is doing the wrong thing, like really wrong thing, they always will have some helper who is a partner in crime, right? So that's what KRAS does. KRAS just stresses the cell so much because of uncontrolled proliferation and other things that it becomes highly dependent on a kinase called TBK1. So in case RAS dependent dependent cancer, it really depends on TBK1. So if you inhibit TBK1, it will die. It is that dependent. Okay. All right. So let's look at it. So we looked at this mutant last time, yesterday, G12C mutant. So this, remember, we discussed the source is activating KRAS. So KRAS, once it is activated, it turns on many signaling pathway. However, this is KRAS dependent cancers are highly dependent on TBK1 driven survival signaling. So if you inhibit TBK1, the cells will die. So this is just showing you RAS signaling pathway, which leads to proliferation, survival, migration, angiogenesis, DNA synthesis. And I showed you this yesterday. Look at the high percentage of KRAS mutants, right? These are, pancreatic cancer is extremely aggressive cancer, and 73% of pancreatic cancer have these mutations. So this TBK1 is a serine threonine kinase. Remember I told you there are three kinds of kinase, serine, threonine, tyrosine, and dual kinases, which can phosphorylate both tyrosine and serine threonine. So TBK1, for our thing, just remember it is involved in cell survival signaling. And KRAS dependent kinases, cancers are highly dependent on TBK for survival signaling, compensatory pathway activation. So if you inhibit TBK1 in KRAS dependent cancer, these cancers will die. These tumors will die. Normal cells will not die, because in normal cells TBK1 is not that important. But in KRAS is the main guy, and it depends on TBK1. So just inhibiting TBK1 will kill those cells. So this is a very good example. KRAS mutant cells require TBK1 for survival. So blocking TBK1 selectively kills KRAS mutant cells. Okay. So this is, you don't have to remember anything, but it's just showing you that it, just look at that, all the pathways. This is not important for you, but look at that. It provides inflammation. Cancer loves inflammation. Okay, it inhibits apoptosis and necroptosis. It promotes proliferation, survival, autophagy. Okay, so this is just summarizing the whole thing, that oncogenic KRAS usually have these mutations, and through RAL-B, it actually becomes highly dependent on TBK1. And TBK1 helps survive through anti-apoptotic pathway and FKB activation. So KRAS mutation depends, it creates oncogenic stress, proteotoxic stress, metabolic stress, and for overcoming all these stress, it depends on TBK1. So if you inhibit TBK1, these cells die. So that is a very good example of synthetic lethality. Now let's look at MTAP deletion in PRMT5 MAT2A. So there are three players here. Okay, so there are three proteins that we have to consider: MTAP, PRMT5, and MAT2A. So I will tell you the functions of each of them, and then we'll see how they crosstalk with each other. So MTAP is methylthioadenosine phosphorylase. Okay, do you guys know what phosphorylase enzymes do? How do you define phosphorylase enzyme? Anyone?
It removes the phosphate?
No, that is phosphatase. Yeah, adding phosphate group, right? So how do you so kinases also add phosphate groups, right? So how do kinase and phosphorylase enzymes differ? Anyone? Probably you guys don't know that, right? So kinase always use ATP to phosphorylate a protein. Phosphorylase use inorganic phosphate to add the phosphate group. That's the biggest difference. Write this down. This is important. Okay. Kinases use ATP to phosphorylate something. Phosphorylase adds inorganic phosphate to add something, to add the phosphate group. Okay. So what happens is that this enzyme MTAP, this is involved in methionine salvage pathways. That means it is involved in the synthesis of methionine. And this is MTA, which is 7-methylthioadenosine. It is converted to methionine in 6 steps. You don't have to remember the 6 steps. I have shown the 6 steps here. So MTAP is used in the first step, and then there are other enzymes. The point is that MTAP is converted to L-methionine, and it is, sorry, not MTAP, MTA is converted to methionine via NTAP. That is the first step of the reaction. Okay, and MTA is 5-methylthioadenosine. So that's one player you have to remember. This is the function, that this is involved in methionine formation. Then there is another enzyme which takes this methionine and it converts it to S-adenosyl methionine, or SAM. So this is the reaction, methionine plus ATP plus water. This enzyme converts to S-adenosyl methionine. Look at this structure here. This is S-adenosyl methionine. Nobody calls it S-adenosyl methionine. We all call it SAM. So this SAM, look at this methyl group, and this sulfur is positively charged. So this is a very good donor of methyl group. So whenever you have to do methylation in cells, this is the donor for methyl group. Okay, and so when it donates methyl group, it becomes S-adenosyl homocysteine, and then it is further converted. We will not go there. So once again, MTAP is involved in methionine formation, MAT2 is involved in SAM formation from methionine. Now PRMT5 is a methyl transferase who uses SAM to put the methyl group on different substrates. Make sense? Now this is PRMT5. So P stands for protein, R stands for which amino acid is denoted by R? Arginine. So this is arginine methyltransferase. Okay, so PRMT5 uses SAM because it is a methyl donor, and look at arginine side chain. It adds the first methyl group, the second methyl group here, right? It could be symmetrical or it could be asymmetrical addition. So now you see three players involved in the same pathway. One guy is involved in methionine formation, the other converts methionine to SAM, and the third uses SAM. Okay, so that's how the three proteins are linked together. So let's look at the synthetic lethality here. So what happens is that MTAP gene is deleted in 15% of all human cancers. Now here is the funny part. MTAP is not a tumor suppressor. So why would tumor cells delete MTAP, right? So actually they are trying to delete CDKN2A, which is a tumor suppressor. MTAP is just next to it. So when tumor cells delete CDKN2A, they accidentally delete MTAP also. So that is like just by being in the proximity, that also gets deleted. So what is the consequence? So this is called passenger deletion. Tumor cells don't want to delete it, they just accidentally delete it. Okay. So now what happens when MTAP is present? MTA is converted to methionine in 6 steps you saw. But when MTAP is not present, MTA is not converted to methionine. But here is the problem. This MTA is an inhibitor of PRMT5. Okay. So when this is not converted, obviously the levels will keep on increasing because it's not being used. So this inhibits PRMT5. So PRMT5 can no longer alkylate arginine, and this is a very important pathway. So by inhibiting this pathway, it is inhibiting it, let's say 80%. So the cells are not really dependent on the 20% activity of PRMT5. Now if you add a PRMT inhibitor to delete that 20% activity also, the cells will die. So if there is MTAP deletion and you inhibit PRMT5, the cells will die. Normal cells have MTAP, you can delete, you can inhibit PRMT5, it doesn't make any difference. But during MTAP deletion, it becomes lethal. Make sense? Or what you can do is that instead of inhibiting PRMT5, you actually inhibit another one. So I just want to repeat this part here. So here it says that high level of MTA act as a natural inhibitor of PRMT5. So then they become highly sensitive. You inhibit that, the cells die. So now cells become addicted to residual PRMT5 activity. So maximum activity is inhibited by MTA. Whatever little is left, the cells are completely addicted to PRMT5 activity, and that's when you inhibit it and the cells will die. Okay. And the other option is that you inhibit MAT2A. So MAT2A is responsible for the synthesis of SAM. If there is no SAM, PRMT5 cannot use it. Basically, it has the same impact. Are you guys following me? So what you do in MTAP deleted cells, MTA levels increases. PRMT5 is inhibited, almost inhibited. A little bit more push, they die. Just a little bit of more PRMT5 inhibition, they die. Or you inhibit this, so there is no SAM. So it's basically killing PRMT5, because without SAM, PRMT5 cannot function. So that is also synthetic lethal. Okay. And now we look at the, so this is exactly the same thing written there. Okay. So indirectly killing MTAP null cells by starving PRMT5 of SAM. Okay. So same thing written in details here. And these are just to tell you that keep in mind, MTAP deletion is accidental, right? They are trying to delete a tumor suppressor. Still it is deleted in glioblastoma, pancreatic cancer, and these other cancers, okay? And these inhibitors are there, okay? So MTAP deletion alone, cells survive. If you inhibit MAT2A or PMT5, somehow they will tolerate it if MTAP is there. But if MTAP deletion is there, you inhibit this, it will lead to cell death. Okay, now the last one. My favorite kinase, Aurora kinase. Okay, so Aurora kinase is present in every cell of our body which is dividing, and it is an essential kinase for mitosis. And if you inhibit Aurora kinase, the cells will die, normal cells will die. So inhibiting Aurora is pretty toxic, but it is overexpressed in every known human cancer. So that is why it is also an extremely important cancer target, right? So this is just showing you it is involved in many different pathways here. Okay, and there are many substrates of Aurora known. So there is a tumor suppressor called retinoblastoma. And if you have to guess, what do tumor cells do what do tumor cells do to tumor suppressors? Whenever there is a tumor suppressor, what would tumor cells do? They will get rid of it. Anything which stops tumors from going, they will get rid of it. That's how poor MTAP gets deleted. It's not even a tumor suppressor, it is just next to a tumor suppressor and gets deleted. So anytime there is a tumor suppressor hanging out, tumor cells will delete it, right? So they delete another tumor. Aurora is a kinase, Aurora is an oncogene, so that is a driver of cancer. So they will not delete that because that is driving cancer. Aurora is the master regulator of cancer, but cancer cells will delete tumor suppressor, and one such tumor suppressor is retinoblastoma protein. So when retinoblastoma protein is deleted, it leads to tumor initiation, proliferation, epithelial to mesenchymal transition. You guys know how bad it is. Genomic instability, it makes cancer stem cell. Remember I told you about cancer stem cell. You give very toxic therapy to a tumor, right? And 99.999999% cells die. Actually one cell lives, just one cell. If that one cell lives, that is the cancer stem cell. And that one cell can form a whole tumor again, a very aggressive tumor, right? So cancer stem cells are very bad, okay? So if retinoblastoma protein is lost, it leads to cancer stem cell formation. So how do we combine Aurora A, which is an oncogene, to retinoblastoma protein? So retinoblastoma protein is obviously a tumor suppressor, and its role is in the cell cycle, and it really helps in the mitotic spindle formation. I am telling you in a very generic way, very simple words. So it is really involved in mitotic spindle formation, okay? And if retinoblastoma is lost, then the whole system is very fragile. Now, as long as Aurora is there, it will take care of the mitotic spindle and the cells will survive. But in such cells where retinoblastoma is missing, if you inhibit Aurora A, they will die immediately. And that is what is synthetic lethality here. Make sense? So that's what it is saying. So primed spindle assembly checkpoint. Okay, so RB1 deficient cells have a hyperactivated or primed SAC. So now they are highly dependent on Aurora A to fix the problem. And if Aurora is even inhibited a little bit, the whole thing will collapse and the cells will die. Okay, and the cells will. So RB loss leads to replication state, chromosomal instability, dependence on Aurora A for mitotic fidelity. If you inhibit Aurora A, the cells will die. Okay, so this is just telling you different drugs of Aurora A, and so these are pretty aggressive cancer, triple negative breast cancer, small cell lung cancer. Okay, almost 100% of these cases have RB1 mutation. So loss of tumor suppressor is a really is almost a hallmark of cancer. Okay, neuroendocrine prostate cancer, remember this is the latest man-made prostate cancer. They also have retinoblastoma, they also lose retinoblastoma protein. Okay, so this is just telling you, so one of the problem with Aurora inhibitors are that they are pretty toxic. So recently this compound was made, Ly compound, and this is 1,000 times more selective for Aurora A than Aurora B. Aurora and Aurora B are like brothers, very similar to each other, not cousins, brothers. Okay, so very similar. So to make an inhibitor which hits only one and not the other is a big thing. Okay, so people think this may get approved by FDA. So this is just showing you, this is the SAC, remember? SAC is spindle assembly checkpoint. So if you have RB loss, this is defective, and now it really depends on Aurora A. If you inhibit Aurora A, completely the whole thing falls apart. So that is what is shown here, Aurora and retinoblastoma synthetic lethality. Okay, everything I told you. All right, so the first lecture ends here. Second lecture and fourth lecture are very long. First and third lecture are short. So let's start the second lecture right away. Okay.